Welcome everyone to the Maddie Revelt's Vision 2024 series of patient and family educational sessions on bone marrow failure syndromes. My name's Jaya Soman. I'm the project manager of the Centre Research Excellence in Bone Marrow Biology here at Maddie's Vision. I'm absolutely excited for tonight's program, so thank you for coming. Before we start, I would like to acknowledge the traditional owners of the land on which we are meeting. I would also like to pay my respects to their elders, past and present, and extend that respect to other Aboriginal and Torres Strait Islanders who are present here today. As many of you know, Maddie's Vision is the only organisation in Australia driving bone marrow failure syndrome research, advocacy, education and support for patients and families. We are the source of support for many through our telehealth nurse service and new peer support program. And we've now supported over 100 patients and families nationally. I invite any of you here tonight who have not connected with these services to please reach out. So in terms of this evening's program, we're thrilled to have two experts in gene editing who will provide an overview of these innovative technologies and the research currently being undertaken in Australia to develop new gene therapies. Following the presentations, we will have a question and answer session, and we really are thankful for all the questions that were submitted before today. And our speakers have endeavoured to cover these in their presentations. But obviously, if you'd like expansion on certain topics, then I'm more than happy to have these answered during the Q&A. Now, absolutely delighted to introduce our first speaker, Dr. Lorna McClellan. So Lorna is a PhD student with the University of Melbourne at St. Vincent's Institute of Medical Research in the Genome Stability Unit, and a clinician paediatric oncologist trained at the Royal Children's in Melbourne and the Evelina Children's in London. I'll hand it over to you, Lorna. Thank you so much.
Thank you, Jaya. I'll just share my screen. Let me know if that looks right for everyone. That's all good?
Yeah,
okay,
great.
Thanks, Andrew. Yes, so thanks so much for the invitation to come and talk to you this evening. So we're going to cover, I'm going to do a bit of an introduction to gene therapies for inherited bone marrow failure disorders. And then I'll hand over to Andrew, who's going to talk in a bit more depth about the type of gene editing that we do in our lab. And thank you, Jaya, for the kind introduction. I'm a paediatric oncologist. I work in leukemia and bone marrow transplant at the Royal Children's Hospital in Melbourne. And I'm also a PhD candidate. I work with Andrew as my supervisor across the MCRI and also St. Vincent's Institute. And we're really interested in investigating novel gene editing technologies, particularly for Fanconi anemia patients. So this is the contents of my talk. I'll talk to you, and we'll go back to the basics of changes in our DNA. We'll talk a little bit about the heterogeneity of bone marrow failure disorders and the different groups of disorders that, that we can see in bone marrow failure. And then I'll introduce gene therapy to you and talk a little bit about the clinical applications of gene therapy. So this is our team. We're the Genome Stability Unit at St. Vincent's Institute of Medical Research in Melbourne, and Andrew, who will talk next, is the head of our unit. And our lab have a particular focus and interest in bone marrow failure, but specifically Fanconi anemia, which is the most common inherited bone marrow failure disorder. And our group within our lab are particularly focused on investigating gene editing technologies, and we're looking to correct genetic mutations of Fanconi anemia. So just going back to basics, so thinking back to our DNA. So our DNA, or deoxyribonucleic acid, are molecules within our body that contain the unique genetic codes that make us each individuals. Our body's genetic material is stored up in 23 paired chromosomes, which are inside the nucleus of our cells. Small segments of chromosomes are called genes, and each person will get 2 copies of each gene, one from each parent. Humans have around 20,000 genes, and genes are made up of a series of letters called base pairs. All of the DNA in each individual is called the human genome, And this is the instruction manual that can control everything in a person, from the color of your hair to your height to the predisposition to certain diseases. Now we're looking a bit more closely at the DNA base pairs. And DNA holds the instructions for all the genetic information. This is then made into RNA, which allows those instructions to be used to make proteins. Proteins are the building blocks within our bodies, which allow our bodies to carry out all the normal processes which are required for day-to-day life. Small changes in the DNA can change the proteins produced, and even a small change in the DNA can lead to changes in the proteins which have big effects in the body's functions. These DNA changes can lead to problems, either with not being able to make a critical protein that the body needs, either it's non-functional or it's made in a reduced amount, or it can lead to building up a protein in the body that's harmful in some way. We've become really good over time at reading the DNA and picking up these small changes that might be causing effects on the human body, but we're discovering new variants or changes in the human genome all the time. We're able to identify these changes because we have databases full of the genetic information from healthy individuals to compare each new test sample with. Any small change can be identified as different or a variant from the norm. Some variations will have no effects on the body, but others can be associated with causing diseases. And we know that around 10% of human diseases are caused by genetic mutations. For individuals with bone marrow failure, some of these tests can be done on the bone marrow itself, but we'll often want to correlate that with another sample from the body, such as a small sample of skin cells. This helps us understand whether any DNA changes are present either only in the bone marrow or whether they're also present in all of the cells of the body and that they might have been inherited. The way that the bone marrow functions and produces cells is also controlled by the DNA, and we'll see this a little bit later on. Our young patients with bone marrow failure will often present to us with symptoms of having low numbers of blood cells. If they have low platelets, they might have bleeding and bruising and petechial rashes. If they have low red cells and anemia, they may be pale or tire easily, or even be short of breath. And they also may have low white cells, and this leads to an increased risk of infections. And there are many different underlying causes as to why the bone marrow fails, which can be quite a diagnostic challenge initially, and it takes some time to have all the important information back that we need to make clinical decisions. There are overlapping groups of disorders which can present very similarly in our patients. The causes can generally be grouped into inherited causes and acquired. Aplastic anemia and myelodysplastic syndrome are acquired, and inherited bone marrow failure disorders are a large group of different inherited disorders that predispose a person to bone marrow failure. Some of the syndromes associated with bone marrow failure we've known about for some time, such as Fanconi anemia, dyskeratosis congenita, Shwachman-Diamond syndrome, and Diamond-Blackfan anemia. However, because of the improvements in genetic testing, we're now aware of many more inherited genes that can be associated with bone marrow failure. In our lab, we're particularly interested in one of the inherited bone marrow failure disorders, Fanconi. This is a rare condition overall, but it is the most common of the inherited bone marrow failure syndromes, and it's caused by 23 different known genes. It's caused by a problem in the way that the cells repair normal DNA damage from everyday life. If the damage in cells builds up and can't be repaired properly, it leads to problems. It's a disorder that can affect many different organs as well as the bone marrow, and patients can present with some of these issues or otherwise around the age of 7 with bone marrow failure. This type of bone marrow failure also runs the risk of developing into leukemia, and if our patients are transfusion dependent, they are treated for their bone marrow failure with a bone marrow transplant. A bone marrow transplant is a treatment which can replace the failing bone marrow, but it's important to remember that we're only treating the bone marrow compartment with a transplant, not the other organs that can be affected in a condition like Fanconi. A bone marrow transplant requires a match to be found for the patient, conditioning chemotherapy that is tailored for the condition and also for the patient-related factors, and this chemotherapy makes room within the bone marrow for the new cells to grow. Cells are then transfused to our patient, and we wait for them to engraft. But there can be complications of a transplant. These include infections, graft versus host disease, sometimes nutritional issues, and there can be early complications related to small blood vessel inflammation. Transplant will always be an incredibly important cure for many of our patients, but it does have side effects that we're unable to avoid. We want to investigate alternative therapies to having a bone marrow transplant. And a natural example of why this might work for Fanconi anemia patients specifically is a group of patients called somatic mosaics, where they have a Fanconi anemia genetic mutation, but they also spontaneously develop a second mutation in a blood stem cell that enables it to revert its function to normal. This normally functioning stem cell can then reproduce itself and outgrow the originally mutated Fanconi anemia cells and repopulate the bone marrow with working cells. What it means practically is these patients have a later onset of bone marrow failure. They have less chance of needing a bone marrow transplant, and they also live longer. Now, gene therapy is being investigated as an alternative to bone marrow transplant for some individuals with Fanconi anemia. Gene therapy is the use of genetic material to treat a disease. Usually what we do is use a healthy copy of an otherwise mutated gene, which will be delivered into a blood stem cell of a patient. For the specialized gene therapies to be able to enter the cells we're trying to target, there are special methods used. And in the case of gene therapy, we use inactivated viruses, and they're used like an envelope to deliver the gene therapy into a cell. This is because viruses are really good at getting into cells, but they don't cause infections because they've been modified in such a way that they're inactivated. A common virus which is used for delivery is lentivirus. There are two different types of gene therapy. In vivo, this means that this is given directly into an individual by means like an injection, or ex vivo, which is what we're discussing here. This is where the cells are collected, treated with a gene therapy outside the body, and then returned to an individual. For bone marrow failure disorders, we're investigating currently ex vivo gene therapies. The process of gene therapy looks a little bit like this schema. So an individual would have their stem cells collected, and that's through a venous line by a process called apheresis. The cells are then processed in a manufacturing facility and treated with the gene therapy. Once it's been confirmed that the cells have been successfully genetically corrected, the patient may or may not require conditioning chemotherapy, dependent on the protocol. For Fanconi anemia patients, the protocol doesn't require any conditioning chemotherapy. This is because of the special advantage that gene-corrected cells have in Fanconi anemia to outgrow the non-corrected cells. The stem cells are then transfused back to the patient, and then they have a recovery period in the hospital where they wait for the cells to engraft and grow, and they're closely monitored and protected from infection. So how does this differ and compare to having a bone marrow transplant? Well, the main point of difference would be that gene therapies use the patient's own cells and not another person's. This means that we don't have to find a donor for the individual. We don't need to use immunosuppression, and we don't have the risk of graft versus host disease, which is one of the major complications that we have after a bone marrow transplant. But gene therapies aren't available for all inherited disorders, and much research is ongoing in this area. Also, different gene therapies are required for different genetic mutations. It's not a one-size-fits-all. In some conditions, only one gene is known to cause the disease. However, in Fanconi anemia, for example, there are many different genes causing the same condition, which mean that gene therapies alone aren't the best strategy. And Andrew will talk to you in a bit more detail about some of the newer strategies. It's important to understand as well that accessing gene therapies can be an issue. Many of these are still in clinical trial form, which means they're not freely available. And clinical trials in gene therapy can also exclude certain patients, especially those that have a well-matched bone marrow transplant donor. And this is generally because many pediatric conditions where patients have a well-matched bone marrow transplant donor have excellent outcomes, and it wouldn't be ethical to use a clinical trial therapy as an alternative to this. So there has been a phase 1 trial for Fanconi anemia A patients. The sites of this were in Madrid and Stanford. They were determining the therapeutic efficacy of using a lentiviral vector carrying the therapeutic FANK gene, and the inclusion criteria I've shown on the slide here. What they did was use the patient's own cells. They mobilized using two products, GCSF and plerixafor. The cells were then transduced with the lentiviral vector and given back to the patient fresh without being frozen and without any conditioning chemotherapy. And again, that's because of the growth advantage that we've seen gene-corrected cells have in Fanconi anemia. The results of the phase 1 trial were that the patients were shown to have engraftment of gene-corrected cells, improvements in cell counts, and those cells were able to repopulate the bone marrow. Because of this, there is now an ongoing phase 2 trial which is underway, and this will give us longer-term follow-up information about the efficacy of the gene therapy in Fanconi anemia A patients, and also the cell and immune reconstitution data. But some gene therapies have had more time in development and are more advanced than others. For example, pediatric inborn errors of immunity is where gene therapy was originally developed. And this initiated about 30 years ago, where the first trials in severe combined immunodeficiency opened. There are now market-approved products for severe combined immunodeficiency in Europe in ADA-SCID. However, gaining access for our patients in Australia has been an ongoing hurdle. Partly this is because of geographical issues, and the enrollments are often required at the site of the trial, but also because of other financial and logistical reasons. It was only very recently that we opened the first ex vivo gene therapy clinical trial in pediatrics in Australia at the Royal Children's Hospital, and this is for a type of severe combined immunodeficiency called RAG1. There are a range of different inherited conditions that gene therapy products have already been developed for. Some are in vivo, directly to the patient, and some are ex vivo, as we've been talking about here. Currently, the open gene therapy clinical trials in Australia I've shown on the slide. As you can see, there's only one open ex vivo gene therapy clinical trial in Australia, but there's a lot of work ongoing through many different groups to provide more access to gene therapy clinical trials in our community and to build the infrastructure to allow us to eventually develop and design our own Australian-based gene therapy clinical trials. And I know it will be of much interest to think about the status of gene therapy clinical trials for other inherited bone marrow failure disorders. Many of these are still in the stages of preclinical work, so still in the laboratory. However, there is published work in both Diamond-Blackfan anemia, Shwachman-Diamond syndrome, and short telomere syndrome, so telomere biology disorders, all using gene therapy or gene editing approaches. And all of this work is really critically important in strengthening the evidence before planning any clinical trials. In Diamond-Blackfan anemia, there is a preclinical lentiviral gene therapy product that's been developed by the same group who ran the successful Fanconi anemia gene therapy clinical trial. But currently there are no active clinical trials in humans in the other inherited bone marrow failure disorders. So with that, I'd like to say thank you very much for listening. Our group in the cancer therapies unit are also investigating improvements in the care of children with bone marrow transplant. And if you've been affected and you'd like to be involved in our ongoing research, there's a short online survey, so please do get involved if you'd like to share your views. And I'll hand over to Andrew.
Thank you so much, Lorna. That was an amazing presentation, really clear overview of gene therapy. And yeah, now I'd like to hand over to Associate Professor Andrew Deans. As Lorna mentioned, Andrew is the head of the Genome Stability Unit at St Vincent's Institute in Melbourne. Over to you, Andrew.
Thanks, and thanks, Lorna, for a great introduction there. You've I can shortcut a few things on my presentation. I'm just going to quickly pop that up. Okay, is that coming through okay? Yep.
Yes.
So welcome everyone tonight, and thanks for coming along. It's always great to present our work and discuss potential in this exciting field with people who are directly affected. So I'm going to be talking about gene editing for Fanconi anemia, but also for other bone marrow failure syndromes. I'm really open to any questions you might have. So Lorna will be the same. Please put anything into the chat that you want to ask us. There are no silly questions. We're just here to try and teach you as much as possible tonight. So please think of anything while we're presenting. So I want to start by acknowledging the team that works in my lab at St Vincent's Institute in Melbourne. And basically we've got two teams within the lab. One is understand the basic biology of DNA repair, and the other is looking at development of therapeutic gene editing. And the intersection of these two areas of research is really Fanconi anemia. So Lorna's already given a fantastic introduction to both current and emerging therapies for Fanconi anemia, but I'll just briefly reintroduce them. So most patients are diagnosed quite young, often almost always as children. But usually treatment doesn't begin in terms of a bone marrow transplant until certain signs are starting to be seen in terms of progression towards bone marrow failure, and that's when a transplant might be initiated. However, for gene therapies and another technology of gene editing that I'll be talking about tonight, what we're looking at is actually starting therapy as close to diagnosis as possible. And that's because we want to be working when there are still blood stem cells available. And obviously with bone marrow failure, those are the cells that deplete over time. So if we're looking to do gene therapy or gene editing, we're going to be working with children predominantly, because we're going to be working close to diagnosis. And that's why Lorna, who works at the Children's Hospital, and the team that she works with, is so important to our work. And Lorna's also pointed out how important it is, this idea that you're going to be transplanted with your own cells rather than cells from a related donor. So in terms of an analogy for how gene editing works versus gene therapy, we like to describe it as similar to like a search and replace in, if everyone's pretty familiar with using Word documents, we go in and we can search for a particular sequence. In this case, we're looking through DNA sequences rather than through a document that's describing our day-to-day work. But then we're able to replace that with a new DNA sequence, leading to new genetic information being encoded. So in terms of when we're treating genetic disorders, we're talking about fixing genetic mutations which are identified through gene reading. So the big kind of game changer, I guess, for this search and replace type of technology came about in 2013 with the publication from Jennifer Doudna and Emmanuel Charpentier's labs, showing the identification of this enzyme called Cas9. Now, Cas9 originates from bacteria. It's a bacterial protein that's normally involved in the way that bacteria become immune to viruses themselves. But it's essentially an enzyme which can be targeted by an RNA sequence to cut DNA sequences and therefore destroy invading viruses in bacteria. But what these guys figured out was that you could repurpose this technology and use it as the search and replace tool in this gene editing approach. So Cas9 is effectively the search function, acts as the search function in this search and replace editing mechanism. But for gene editing, so Cas9 will find a sequence in the DNA, and perhaps for those of you not familiar with DNA, it's actually made up of 4 letters. That's why we've got all this kind of random sequence here, is because it represents the 4 letters of the DNA code that encodes, as Lorna introduced, for different enzymes and functions in the genome. But for gene editing, we also need a replace option, and this replace function is conducted by providing an extra piece of DNA to the cells, which act as the template to introduce this new DNA sequence that we want to replace. So one way that we can measure gene editing is to just read the DNA sequence after we've done DNA editing. And Lorna introduced how we use DNA sequencing to identify mutations in cells, but we can use this type of reading to identify whether gene editing's actually worked as well. But one problem with using this approach in the lab is that it can take several weeks to get a result, and several weeks is quite short in terms of a clinical diagnosis, but it's actually really slow when it comes to conducting research. So a postdoc in my lab, Astrid Glasser, invented an assay that can actually measure this search and replace function in the genome that's actually used now by hundreds of labs around the world. And this actually doesn't require us to read the DNA sequence, because instead we can measure whether editing has occurred in cells by looking at a color change that's induced. And so just for the purposes of introduction, I've not put the actual DNA sequence of the green fluorescent protein that we used. I've just called it green. And what happens is that when we edit the green gene, the cells become blue. And so we can read this blue gene by just looking visually at the cells, whether they're green or blue. But of course, because of the way that the CRISPR-Cas9 system works, it's also prone to making mistakes. And if mistakes are made, then the sequence is lost and the cells become dark or grey and we can't see them at all. And so if kind of what this looks like down the microscope, you can see here we've got hundreds of different cells that are growing in a dish when we look at them down the microscope. And when we start off the experiment, they're all green. But when we conduct gene editing, you can see that many of them now become blue. And if we overlay the green and the blue channels, as we call them, onto all of these three pictures together, you can see that there's green cells, blue cells, but there's also a lot of dark cells which have lost their color. So another way that we can count the number of green, the number of blue, and the number of dark cells is by using a method called flow cytometry. You don't have to know the details about it, other than if cells are up here, each dot is a single cell, they're green. If we give a Cas9 enzyme which targets this green gene, you can see many of the cells lose their greenness. But if we give a template for this blue gene, the cells now become blue. So what's important to take home is that we get really good levels of editing using this Cas9 technology, 23% cells in this experiment, but actually still a lot of the cells are edited incorrectly and they become dark. So CRISPR-Cas9 was only discovered in 2013, or its ability to be used as a gene editing tool. But in 2019, this new development of CRISPR-Cas9 called prime editing was introduced. And the scientific version of prime editing here is that it incorporates a reverse transcriptase enzyme and an RNA-encoded template for the genetic change. But what this means for gene editing is that we've gone from this kind of pretty indiscriminate editing mechanism, which I've characterized here, to something that's much more efficient and works all of the time. And what you can see here now, when we use prime editing for exactly the same assay, we get about 50% of the cells that are properly edited and only around 1% of them that are wrongly edited. So I often get asked, when will gene editing be ready for patients? Well, with CRISPR-Cas9 and for many diseases, it's actually already in clinical trials. And this is one review from a couple of years ago showing how CRISPR-Cas9 has been used for various different cancers, genetic diseases, virus treatment. And then this is a slightly more recent one, which is looking specifically in blood tissues, and you can see that there's many different CRISPR-based technologies that are already going into clinical trials in patients. But when will gene editing be ready for bone marrow failure syndromes? So I've put down here like a list of the 5 key stages of developing a gene editing technology for use in for patients. And really, if we now look at the progress we've made against all of these requirements, what you can see is that the refinement of these editing techniques has been improved dramatically, even in the 5 years since prime editing was discovered. And we can actually now target most, if not all, of the mutations that are causative in Fanconi anemia and other gene disorders. So progress has been rapid and really, really exciting in these first two steps. Then as we move down the list, we also need to realize that we have to get these editing tools inside of blood cells so that we can actually induce correction of the genetic causes of the syndromes. Lorna introduced earlier that we can use viruses for this. It's much harder with CRISPR and prime editing technologies to use viruses just because they're very large genes that we have to deliver. However, there are other approaches that I'm going to talk about in a moment that have been developed. Then there requires extensive preclinical testing because we need to use animal models and human cell models in order to show the safety and efficacy of the technology. And then finally, probably the bit that we're still lagging behind in is in clinical trials and in regulatory approval. And this is where the biggest advances are going to be made over the next couple of years, just as the technology really gets, as all of these other hurdles are going to be overcome. So I'm just going to talk a little bit briefly about these two parts, which we've worked on quite significantly over the last couple of years. And the first of which is how we actually deliver these gene editing tools into blood cells. So we've got something that works really well in the laboratory. How can we actually transfer that into something that's going to work in a clinical setting? Now, we like to work with these cells that grow, they're immortal, they grow forever, they're very easy to culture in the lab, and we can do gene editing using all of these different delivery methods. You don't have to be familiar with what they are, but the main one and the easiest one that people work with is DNA. So PE is prime editing. We heard about viral delivery. I'm going to talk about messenger RNA a little bit, and lentiviruses as well we heard about. But when it comes to blood stem cells, the ones that are easiest to work with in the lab are basically a complete no-go in trying to edit blood stem cells, which is really what we're interested in. So what we've focused on are these last three, and really to develop a preclinical model for applying gene editing in blood stem cells. We've worked with a couple of other labs at St. Vincent's Institute, primarily Jörg Heilhorst, to make mice that express this green fluorescent protein. This is a cartoon, but this is actually what they look like when they're young. They look like they glow in the dark, but in fact, they only glow when we put them onto a special blue light source. But what we found is that the blood cells from these mice, when we isolate them using, again, specialized techniques for isolating blood stem cells, that they're 100% or 99% green, the blood stem cells. So we've got green mice with green blood stem cells that we can actually look to see whether our gene editing is working in these primary non-lab cells. And Lu, who was a PhD student working in the lab up until last year, worked extensively with the primary hematopoietic stem cells or blood stem cells from these mice. And I think I've shown to this group recently some of these exciting results where we were getting gene editing of up to 37% of the cells becoming blue, and that these cells can differentiate and become normal blood cells in the lab. I also wanted to just quickly point out this messenger RNA delivery technology that we're working with a group at Monash University who's been leading the development of COVID vaccination, COVID vaccines. And you've probably heard about this technology because we've all had a shot in the arm now of a COVID vaccine. And these vaccines use messenger RNA to deliver a COVID protein into cells of our body, which our cells then express. And we make antibodies against the COVID S protein. That makes us all less susceptible to the COVID virus. But we can actually use exactly the same technology to deliver these gene editing reagents inside these lipid nanoparticles based as RNA molecules. And we've got some pretty good evidence that they work in human cells and mouse cells that way as well. So this is some work from actually from Lorna, showing now that we've now got this editing working in human cells. And Lorna has been working with Andrea Elefanti and Elizabeth Ing's lab at the Murdoch Institute, where they've able to culture these hematopoietic stem cells outside the body, and she can edit these into high levels of blue cells. And very soon we're going to be transplanting these into mice to show that the blue cells can still become normal blood. And I'm pretty confident that they will, because they do in vitro as well. We're not just interested in editing green to blue. We have also been testing out various reagents for editing Fanconi anemia genes. For these, we have to read the DNA sequence, and this is just an example of what one of those DNA reads look like. We read it using coloured bases, which are read as green, white, blue, or red. And what you can see is for this specific FANCF mutation here, we can get editing of about 33% of the reads are edited very specifically at this DNA sequence to change it from an from a C to an A. Okay, so that's the technology. I'm running out of time, but I did just want to briefly introduce one small aspect relating to the ethical considerations around gene editing, because it's something that is often raised with me when I tell people that I work in gene editing. You might have seen some of the titles or some of these type of magazine covers about how gene editing is going to potentially lead to eugenics or changes to eye colors or inherited properties. And actually, there have already been babies generated in China using CRISPR gene editing. And these, there was a big backlash when this occurred back in 2018, and I don't believe it's happened subsequent to this. However, what we're talking about here is gene editing for bone marrow failure syndromes, and the editing that we're doing is in mature blood stem cells. It's not from embryos, not from embryonic stem cells. And this approach is not transmissible to the next generation. So we're not going to be creating mutants or altering the human evolution. So really the ethical considerations that Lorna briefly touched on are going to be more around the cost of this technology, but also who's going to get access to begin with. We've already talked about how some of the clinical trials are operating predominantly over in a few countries. How can we actually make sure that we get access sooner rather than later in Australia, and also making sure it's safe for treating as many people as possible. So finally, I just want to briefly touch on a large consortium that I'm part of, or actually leading within Australia, which is called Page Turner, which pioneering the application of gene editing to bone marrow transplant using RNA. And really what this team of both clinicians and gene editing researchers and people with experts in mRNA delivery technology, really what we're trying to do is push all of the technologies so that we can be ready for a clinical trial within a reasonably near term in the future. And for the future, in terms of preparing for an Australian trial of gene editing in Fanconi anemia, Lorna has been working closely with Rachel on establishing Australian trials for SCID at the Children's Hospital, and really a lot of the work that's gone into that hopefully means that Fanconi anemia might be one of the next ones to follow on. We're also, or I'm also part of an international consortium with several groups, including the original discoverer of prime editing, that are at the forefront of developing these technologies for application in bone marrow failure syndromes. So yeah, that's all I'm going to be talking about.
So thank you everyone for attending our session tonight. We really hope it's been informative and helpful to you and your families. Huge thank you to Andrew and Lorna for your fantastic presentations. I've certainly learned a lot, and I'm sure that everyone else here tonight has as well. And it's very exciting to see what the the future will bring. We will be sending a short survey to everyone tomorrow morning, so we'd really appreciate your feedback. It does help us to inform future sessions and how we can improve. Tonight was the third of 4 sessions for this year, so please do look out for details of the 4th session in November, which will be more an allied health nursing focus. So that does conclude the formalities for the evening. I do thank you so much again, and wish you a good evening.